Today we will learn about N-Glycosylation. So before we start, I would like to point out my mistake in the previous video is that I have linked uh, polypeptide chain and this N-glycan through this uh, glucose through this glucose. However, it is not the case. Actual link happens like this. This is the sequence asparagine, any amino acid except proline or except proline, and then this is serine and threonine, and it should bind with this N-acetylglucosamine. Okay, so now let's start our topic, that is N-glycosylation. So N-glycosylation start even before the translation of the polypeptide chain occurs. So what happens is that initially this Dolichol diphosphate hydrocarbon chain is usually found in the membrane of endoplasmic reticulum. And what happens is this part is the cytosol, and this is the ER lumen. So first in the cytosol, this addition of in this Dolichol diphosphate hydrocarbon chain, which is usually found in the endoplasmic reticulum, this N-acetylglucosamine, 2-N-acetylglucosamine is added. It is usually added with the help of UDP, nucleoside. That is UDP comes with this, and it reacts with transmembrane. It reacts with endoplasmic reticulum membrane and forms UTP and releases this N-acetylglucosamine to the diphosphate this Dolichol chain, diphosphate Dolichol chain, and UDP is very much flexible. It can carry N-acetylglucosamine, mannose, fucose, and some other monosaccharide. However, and after the addition of the 2-N-acetyl glucosamine, addition of 4 mannose occurred in the cytosol part. So this usually occurred with the help of GDP. GDP come and react with ER membrane, come and react with ER membrane to form GTP, and it has mannose in it, and it will release that mannose that will link to the this glycan. It will link to this N-acetylglucosamine, and 4 of this mannose link above the in the cytosol. After that, this Dolichol chain, will flip inside the ER lumen, and inside ER lumen further addition of mannose occur, and this time the mannose addition occur is with the help of this another Dolichol chain with single phosphate, and it contains the it has this mannose, and as we have seen that GDP will react to give GDP. So when GDP come and react with this Dolichol single phosphate chain, it releases this mannose, and it will bind to the this glycan structure. GDP come and react to form GDP, and GDP carries 2 kind of monosaccharide, either mannose or fucose, and when this addition of mannose will continue till it acquire this kind of structure. It has like 9 mannose and 2 N-acetylglucosamine. After that, we need to add the required glucose, 3 glucose in this. 3 glucose are added in the similar manner the mannose is added, that is the Dolichol chain with single phosphate will have this glucose structure, and when UDP react with react with this Dolichol single phosphate chain, it form UTP and helps in release of this glucose to bind to the one end of the mannose, and this process continued till it acquired 3 glucose in it. We need to understand that in this representation I have represented N-glycosamine and glucose with looks like N-glycosamine and glucose in a similar form. However, they are very different from each other. We need to understand that. In this way, this glycan structure will form, and it will continue to remain in ER lumen until it gets a polypeptide chain with this sequence. It will wait for the polypeptide chain to start translated. It will wait for the polypeptide chain to be present in the ER lumen, and this process is triggered without even the translation of the polypeptide chain starts. So this is the initial step for N-glycosylation. So later on we will study how this N-glycosylation carried forward in ER lumen and then to the Golgi. Okay, thank you.